Hello, my name is Julieta Rey, and in this video, I will be explaining DNA replication, the leading and lagging strands, and the enzymes that are used to make this process work. I will also be talking about telomeres. DNA is first made out of phosphate groups, nucleotides, and deoxyribulose sugar. Nucleotides include adenine, guanine, thymine, and cytosine. Adenine and guanine complement each other, while cytosine and thymine are together. I will first talk about prokaryotic replication, then I will later implement eukaryotic replication with the telomeres. As I was explaining before, this is a good picture to emphasize the nitrogenous bases, deoxyphosphate groups, and deoxyribose backbone. So as I said before, here are the phosphate groups at the end. This is the deoxyribulose sugar, and these are the nitrogenous bases. So as I say, adenine, thymine go together, cytosine and guanine go together. DNA first runs from the 5' end to 3' end, and on the other side it runs from 3 to 5' end, which we will later get into how important that is. I would first like to emphasize that DNA replication can happen on any origin along the DNA. These bubbles is what starts a DNA replication, and they're called forks because as you can see it looks like a fork road, and that's where the DNA replication starts. So what I would first like to introduce is the enzyme helicase, which is right here, and it is an enzyme that separates the two strands, the leading and the lagging, and after this enzyme separates, that's when it starts to replicate. So as you can say, the leading strand is a faster process. It replicates faster than the lagging strand just because it's going from a 5' to 3', while the lagging strand goes from 3 to 5. So it lags a little bit more and takes more time, but I will definitely get into that later in the video. So what I would first also like to introduce is DNA polymerase, which is right here. So after helicase comes in and does its job by separating the two strands, DNA polymerase is also an enzyme that adds the nitrogenous bases to where it's supposed to belong. So as I stated before, it's adenine and thymine and cytosine and guanine. So that's what DNA polymerase does. I would also like to introduce these purple proteins right here. They're binding proteins, and they keep the strands so stable and separate from each other so that they don't intertwine and it just becomes a huge mess. So that's what those proteins are there for. Polymerase can only add new nitrogenous bases from the 3' end. So it goes this way. It cannot go the opposite way, and I will later explain that happens in the lagging strand, and that's why it tends to be slower than the leading strand. I would also like to introduce topoisomerase, which is an enzyme that relieves the overwinding strain that the DNA is putting itself through, and it's right before the fork, the replication fork, right before helicase goes in and does its job. DNA primase, which adds RNA primer to the nitrogenous bases. Once the RNA primer is set into the nitrogenous bases, then DNA polymerase will go into its job and convert it to DNA. And the only difference is that going with thymine, it will go with uracil. Then DNA polymerase adds the thymine. Because RNA is here and then here it's automatic, like just recently DNA, RNA and DNA can't. So they run into each other. And so the enzyme DNA ligase, which is right here, it will clean up this mess, and the DNA polymerase will able to continue this job. So ligase is kind of like, I like to think of it as a glue. So it glues the two strands together by cleaning up all these disjunctions and messy thing between the RNA and the DNA. And then we'll replace the uracil RNA. So now from one strand, there will be 2 copies of DNA. This is a prokaryotic one. And right now I will explain the eukaryotic part with the telomers. Thank you for watching this video. I hope it was very informational. So what telomerase and telomers do, it's known as a garbage DNA. It puts sacrificial chunk garbage DNA in the replication process. So every time it divides, it loses a piece of it, and it keeps putting more and more junk on the ends of the DNA, so you could divide and divide forever. But then it would later die, and you can't have that gene or protein that you're going to need ever again. So with aging, of course, every time it divides, you can only have so many divisions. So that's why aging happens, and cancer.